this event is the following. I will take the lead here on a few words about our company. Then Stefan will take over with insights into the compound libraries, and afterwards Ben will continue with HTS technologies and workflows and the case study, and afterwards we will reassemble again for a live Q&A session. So I will start with a few words for those of you who haven't worked with Nuvisan before. We are an integrated CRO with more than 40 years of track record in Europe and also worldwide. We take pride in seeing ourselves as a science CRO. We have extensive industry experience thanks to the fact that many of our sites have been acquired from pharma companies, and above all, science first and client focus is important to everything we do. Looking back a bit into our history, we have been around for more than 40 years. We had an intense acquisition phase between 2010 and 2020, where we have taken over a lot of pharma sites. The most significant acquisition has happened in 2020 with Innovation Campus Berlin, where we have taken over 380 people from Bayer Pharma. And then we had two very exciting events in the last few years. In 2021, we partnered with ALS. And then very recently in April, ALS took full ownership of Nuvisan. So we are now part of a much bigger family, and that puts our numbers a bit into perspective. We started off with a very small team. We had acquisitions of dozens and hundreds of people, and before the big ALS decision, we were in Nuvisan around 1,000 people. So now we are part of a family of 19,000 people present in more than 70 countries with more than 350 offices, and beyond the CRO industry, also in other industries. So these are exciting times for us here at Nuvisan, and we are very positive looking into the future together with our ALS colleagues. Now, zooming back into what we do at Nuvisan, our focus is drug discovery and drug development. And as I already said, we are an integrated CRO covering everything between concept stage, which is target discovery and target validation, all the way into the clinic. Here in Berlin, we are a drug discovery operation. So our portfolio in Berlin is in vitro, in vivo pharmacology, is discovery bioscience, is HTS, is computational and medicinal chemistry, as well as DMPK and toxicology. And then if projects move further in the drug development pipeline, then we are happily collaborating with our colleagues from the other Nuvisan sites on chemical development, on toxicology, formulation development, and eventually also clinical studies. But in the context of today's webinar, we are not talking about clinical studies, but very much about the HTS screening part. And with that brief introduction, I wanted to do it a bit more interactive, and we have a little poll prepared for you to learn more about you. And the question is, have you ever worked with Nuvisan before? And can I start the call here? I kindly ask you to please participate by just clicking on one of the three options. We let this run for a while. Thank you very much for being so active. And it already is clear. It's a good mix with a slight advantage for those who have really never worked for us with us. And so I think these are optimal conditions for today's webinar, which is designed such that you learn a bit more about what we do, and the emphasis today is on HTS screening. So I thank you very much for participating in that call. And with that, I think I pass the microphone on to my dear colleague Stefan.
Yes, thank you very much. Let's get started with an overview on the libraries that we offer here at Nuvisan. First of all, on the left part of the chart, you see the Ultra family that consists out of the Ultra HTS stack, the Diversity HTS stack, the Engli, and the Goldrand, which are all part of the sharing Bayer pharma legacy. Altogether, 3 million compounds of a really high chemical diversity and a high percentage of unique chemical structures with a high proprietariness. On the other hand, we have the Explorer library, which is a very new library. We purchased it at the beginning of this year, and it's of a commercial origin, designed really to have a high diversity with lead-like properties, and we have our fragment library with 2,000 compounds. For the Ultra family, let me just go a bit more into detail on the Engli. That's a subset that's screened in addition to the other sets. Its focus is new IP starting points and really good physical properties. The Goldrand subset is a library that was cherry-picked and curated for special purposes. If for technical reasons, for example, you want to go for a smaller number of compounds, if you need a higher concentration, or if for other reasons you just wanted to screen a smaller set, but still maintaining the same chemical diversity. On the next slides, I would like to go more into a detail and describe a bit the strengths of each of those libraries and have a look together with you from different angles on different parameters and different properties. Let's get started with the Ultra family. So the sharing Bayer pharma legacy library of around 3 million compounds. Those compounds have been built in the last decades based on a lot of pharma projects. They've been screened in many HTS and also used for many virtual screenings. And they have a proven track record to bring compounds into clinical development. And because of this rich heritage of screens, we have a life science database of around 1 billion data points, which are historical HTS data, which are measured PhysChem data, which are PK or pharmacological data. And of course, we also have machine learning-based predicted data for all of those compounds. Overall, a very balanced PhysChem property distribution, high internal structural diversity, and clearly one of the strengths is that we have a lot of unique compounds, especially compared to external collections. One of the questions that we often ask from clients, if those compounds come from pharma heritage, well, then maybe a lot of those compounds are part of patents already. And that's fortunately not the case. More than 95% of those compounds from the Ultra family are IP-free in the sense that they are not an exact match with any of the patents from the patent database. On the right part of the screen, you see what I mentioned with historical HTS data. So those compounds were screened in a lot of HTS campaigns, a majority in more than 50 HTS, a lot of compounds even in more than 100. And 90% of those compounds have been a hit in less than 5% of the cases. So if you run an HTS and you have a high hit rate, that's an opportunity to remove, for example, those who have been hitting frequently, but still the huge majority of compounds is left because they have not been appearing in a lot of compounds. So that's, of course, also a quality sign for this library. Let's switch to the Explorer library. So that's this new high-value commercial collection that we purchased based on lead-like properties, still a significant chemical diversity, and you can see some of the examples structures on this slide to give you just the first impression on this library. The fragment library consists out of 2,000 compounds that we use for biophysical or biochemical fragment screens like TSA, SPR, or NMR and MS. And half of those compounds, 950, they are also part of a subset that we use for X-ray fragment screen. On the next slides, I would like to go more into detail and compare these libraries. For example, the first property we're looking at is a proprietariness. The Ultra already mentioned, more than 80% of the compounds are proprietary. That's this Pharma Legacy library. The Explorer library, on the other hand, compound structures are public. So the advantage is that we can share the chemical structures with you from the beginning on. That, of course, is not possible for the Ultra because otherwise it would not be proprietary. But we are sharing, of course, the chemical structures with you as a license event. And in between, we work with a firewall consultant, which was really successful with all of our clients. Having a look, sorry, I went back. That was not the purpose. Having a look at the physical properties of the Ultra versus the Explorer. Nice drug-like properties for both libraries, a bit more lead-like for the Explorer. For example, molecular weight, the average for the Ultra family around 400. For the Explorer is a bit lower with 330. Log P, C log P, 3.6 for the Ultra, 2.4 for the Explorer. That expresses a bit the different type of chemical structures that you find in those libraries. Having a look at an overlap between the libraries, that's the left chart that you see, and less than 1 per mil of the compounds from our Ultra library is also part of the Explorer library. And looking more at the similarity, taking a Tanimoto similarity coefficient of 0.6, we can say that two-thirds of the Explorer library has no similar molecular match in the Ultra library. So overall, a really orthogonal set of libraries, which also for certain projects might make sense to screen first one, like the Explorer, and go into the Ultra afterwards, because they're so orthogonal. Looking more at a diversity analysis, keeping in mind that the Ultra library is 9 times larger, of course, it covers a larger chemical space, as you can see in blue. Nevertheless, the Explorer in orange still maintains, based on its library size, really significant chemical diversity. And that was also the reason why we picked those compounds for this additional